All right, on to the large molecules found in living things. These are your macromolecules, and you have four of them. You have the carbohydrates, you have the proteins, you have the lipids, and you have the nucleic acids. The nucleic acids, DNA and RNA, is a separate video that we'll make for you on that one. So we'll just keep going here with these macromolecules and all. So the first one are your sugars, and that would be your carbohydrates. So carbohydrates are different levels based upon the arrangement of the molecules. And so when you just have single units, these single ring structures or whatever, they're called monosaccharides. And some examples there, it's not exhaustive, just some examples: glucose, fructose, and galactose. It just so happens they all have the same molecular formula, but they're arranged differently, so they have a different structural formula, which makes them isomers of each other. You can put two of these or more together, and when you do, you produce a di means two, disaccharide, and you lose water, or water is a product when you do that. So that's known as a dehydration, loss of water, synthesis. You can also call them dehydrolysis. Ooh, I don't like that. I'll stick with dehydration synthesis. In the old days, we called them condensation reactions, and also, which means if you go the opposite way, if you take a large molecule and you split it up into smaller ones, you use water. Water is a reactant, and so those are known as hydrolysis. So when water is on the left side of the chemical reaction, it's a hydrolysis. When water shows up on the right side of the chemical reactions, it's a dehydrolysis. When you put the sugars together, there is a bond, a link that forms, and that's known as a glycosidic linkage. You put two together, you get a disaccharide. You put another one in there, you think, well, let's try. Okay, then they go oligosaccharide, and then they'll go all the way up to polysaccharide when you get a whole lot of them, a polysaccharide and such. Polysaccharides, when it's glucose, that's a storage molecule, both in plants and animals. In plants, it's starch. In animals, we call it glycogen. Now, polysaccharides can be for structures, for building things, and then in plants, that's the cellulose and such, and then it goes and makes other things. And in insects and fungi, it's known as chitin. So cellulose, which is a structural polysaccharide, and because it's built and put together like it is, you and I can't digest it. We don't have the enzymes for that. So that actually becomes part of the fiber that we need in our diet. Just certain fungi, certain algae, protozoans, bacteria, certain ones have these enzymes that have this ability to break up the glycosidic linkages in cellulose and consume that. Most of us humans and mammals and such don't. So, well, goats eat plants and cows eat plants. Yes, they do. But they have these specialized microorganisms in their gut that allow them to break it down. Termites eat wood. Yeah, they do. But they have a specialized microorganism in their gut that enables them to get to break it down and get the energy out of it and such. So, and by doing that, they're going through this process known as hydrolysis to do that. I mentioned it a while ago, these other ones, these oligosaccharides. Oligo means few. So the word oli means few. And so you'll see these in energy bars, these athletic bars and such. So they're shorter chain polysaccharides so that your body can break them down and you can get to the sugar more quickly. You need this. We take everything like that and we break it down to the molecule glucose. That's the primary fuel source. We call that blood sugar. Some are able to do it with oxygen. Some are able to do it without oxygen. You have muscle fibers. You have two types of muscle fibers in every muscle, and some are anaerobic and some are aerobic. And also, you take this and your anaerobic ones, which are like for burst of speed or energy and such for strength, they will take that glycogen, that polysaccharide, and break it down very quickly. Your other types of muscles that you use for long distance running or for endurance and such, they have the ability to take glucose more easily and use that and such. It's also stored. This glycogen is stored in your liver, but you also store it in your skeletal muscles. Every one of your cells has these sugar markers on them, and some are linked with fats and some are linked with just by themselves, and they call this your glycocalyx. This is your sugar coating. This is what your immune cells read. And your glycocalyx is different from your neighbors, which is different. It's different from your wife. It's different from your kids. It's different from me. We all have our own unique individual glycocalyx. And our immune systems read that, and if something shows up that's different, they attack it. That's that part of the rejection that you hear about when you do organ transplants. So proteins are polymers of amino acids. Amino acids are the building blocks of proteins. There's only 20 that occur naturally. Plants make them themselves. So we say that they have this ability to feed themselves, so they're autotrophic. You and I can make some, but we have to ingest the others. So we have some that are natural in us, and we have others that are essential, which means we have to take them in our diet. And also, your body can take these amino acids, single amino acids, put them together to make a dipeptide, put them together to make a polypeptide in a dehydrolysis, synthesis and such. So, and then you go from a mono di to a polypeptide up to proteins. And you and I are proteins. And when you look in the mirror, you're looking at proteins. The code on how to string them together, that's the DNA. That's a different video. So proteins exhibit levels, these levels of structures. And all proteins have a primary level of structure. That's the amino acid sequence. When you start putting them together, the elements react and repel and attract, and they form these shapes. And there's two big groups. Some form a coil, if you will, kind of like the old-timey telephone cords that coil up. That's an alpha helix. Some lay out flat like a piece of paper, but it folds like pleats, like you would iron pleats into your pants and such. So they call that a beta pleated sheet. Okay, so that's a secondary level. All proteins have a primary. All proteins have a secondary. Some important proteins that are found in your skin and such have these, like collagen and keratin and elastin. They have like that helical shape to them. Other ones have that pleated sheet to them and such. So you start taking that coil and you keep coiling it, and it will form a glob. That is a tertiary level structure. And these have a three-dimensional shape. A lot of the proteins in your body, and you are a big protein factory. Your runoff of proteins, everything about you is proteins. Enzymes. Enzymes are biological catalysts. They are proteins. They have shapes to them. They have things that they act upon, and only those certain things. So we have a theory that we always work with this, and we call it a lock and key theory. Many locks, many keys, but only one specific key for a specific lock. That's enzymes and how they work on their substrates. What the enzyme works on is substrate. So proteins do a lot of things in the body. They move things, they carry things, they're hormones, antibodies are proteins. And then you think, well, I thought just muscles were proteins. Everything when you get down to it is proteins. The fourth level, which is just a couple of examples, and one example is hemoglobin, which is found in your red blood cells, which is a protein, and it carries oxygen, and that is a quaternary level. So it's like you take these three-dimensional levels, these tertiaries, and you put them together, and they usually have some non-protein, some metallic, some ion in them. And so hemoglobin is an example animal cells. Some lower animals, instead of having iron there, they have copper. Some of your aquatic worms and such, your crustaceans and all, they have a copper there. Plants, they have chlorophyll. Chlorophyll is an example of a quaternary structure, and they have magnesium in the center of that. Lipids are your fats, your oils, your steroids, your phospholipids. These are hydrocarbons. And a hydrocarbon is a long chain of carbons, which is nothing but carbon and hydrogen. That's why they call it a hydrocarbon. Since they don't have an oxygen, they don't look like water, so we call them nonpolar. If something looks like water, we call it polar. If it doesn't look like water, we call it nonpolar. So fats and water don't mix. They just, because it's nonpolar and polar and all. But fats are very useful for the human body because it is a large reserve of energy. Fats are the king of fuels. Those little bonds, that's the energy. These little lines right there, the little electrons, that's the energy, the stored energy and such. So when you're looking at these fats, you look at these bonds. If there's single bonds there, then we say that they are saturated fats. If you see a double bond there, there's an example of a double bond, then that is considered an unsaturated fat. So you've heard a lot about saturated, unsaturated and such in your nutrition and what everybody wants you to eat. So if it's a solid at room temperature, like butter, like bacon grease, that's saturated. If it's a liquid at room temperature, like corn oil, olive oil, and such, that is a unsaturated. So saturated usually have an animal origin. Unsaturated usually have a plant origin. That's one way to think about it. And then the last one we'll mention here, phospholipids. And these are a special type of lipid. They have a round end, which is the phosphate, and that phosphate is polar. So we say that it loves water, so that we say that it is hydrophilic. So it points toward water. The tails, and each one has two little tails, they are hydrophobic. They fear water, so they point away from water. That causes the phospholipids to arrange themselves in a bilayer with the tails pointing toward each other. This sets up and creates the membrane. So when we go to the next video and we see these membranes and such, that's what it's pointing toward, these phospholipid membranes. All right, so there you go. Good to go.